Hey, hey, hey. All right, go ahead and grab for me movement across the membrane. And this is the only one for this part. Okay, so make sure you're writing down additional notes and highlighting some things that I say. Okay, this is all about how particles move in and out of the cell membrane. So if you look at your cell here, we said that your cell membrane is semi-permeable, right? So I said it's choosy. Some things can go in, some things can't. So that's what these notes are all about. What can go in and what can't. So just at the top of your notes, go ahead and write this in for me. Normally, small, uncharged molecules can move in and out. Okay, large or ions, charged particles, we'll say molecules to make it match, can't travel through. So if you're small and uncharged, so you don't have like a positive or a negative charge, normally you can flow freely. If it's large and charged, it can't. It can't just move through the phospholipid bilayer. So the first part says that CO2 and oxygen, they're both small and uncharged. They can diffuse easily. Ions or charged particles and water have to move through protein channels, like they have to go through a protein. Okay, passive transport, there's two kinds. There's really three kinds, but two kinds on here. Oh, there's three. I'm sorry. I'm just crazy. Okay, there's three kinds. There's osmosis, which is the movement of water. There's diffusion, which is the movement of not water, like CO2 and oxygen. And then there's facilitated diffusion, which is just diffusion, but it goes through a protein channel. So osmosis and diffusion both go through the phospholipids. Okay, so we're starting with diffusion. Diffusion, this is the process where it's going with a concentration gradient. So if you look at the picture, it'll show you there's a whole bunch of the yellow molecules on the left side. So this is high concentration. Well, they're going to move to a low concentration. So all passive transport moves from high concentration to low concentration until equilibrium is reached, or homeostasis. Okay, so for example, let's draw this out. If here's your cell, and you have a whole bunch of oxygen molecules out here, and you only have one oxygen molecule on the inside, the oxygen molecules are going to move from a high concentration to the low concentration until they reach equilibrium, until there's an equal amount on both sides. So until there is 4 and 4. And remember, this didn't take any energy. There was no battery. I didn't have to plug it in. There was no ATP. This was passive transport. Passive transport means no energy. And really, it's the exact same thing for water. And so I could draw it out. But water moves just like that, high concentration to low concentration. Water always moves from hypotonic, hypo means low, to hypertonic, high concentration, until it reaches isotonic, which means equal. So write that in above that. Hypo means low. Hyper means above. And iso means equal. But water is very important, so we should be drinking it, yes? You can see this is actually called a U-tube. Ironic, I know. But the water is going to move towards the the solute. So you can see the blue is the water, not the purple dots, but the blue. You can see the water moves to the right side. Okay, but water has to be regulated because it's so important. So pure water versus normal water, pure water is always the hypotonic. So if something is turgid, like think about the plant cells in there, if something is turgid, that means it's swollen. There's a lot of water. If you bite into a piece of celery and it's crunchy, that's turgid. Flaccid is going to be limp, like floppy. This refers to a condition when there is not enough water. So nobody wants to eat lettuce that is flaccid. And then plasmolysis is where there's so little water, the cell membrane, which is green in the picture, has actually pulled away from the cell wall. So those are three different conditions. Turgid, lots of water. Flaccid, not enough water. And then plasmolysis is actually where it's dehydrating, it's pulling away. Okay, water potential, which is represented with this Greek symbol that kind of looks like a fork, like that. Okay, water potential is really just how bad does the water want to move. We know that solutes like salt absorb water. So if you eat a lot of salt, your body will store a lot of water. And so water wants to move in different ways. So pressure can make water want to move, like the picture. We state that water is moving from high potential to low potential. And so write this in for me, this formula I'm going to give you. Okay, water potential, total water potential is equal to pressure plus the solute. And the solute number is always negative. And we may see some problems where you just fill in the math and have to add it up. Okay, and then facilitated diffusion is just like regular diffusion, which you see on the left, but it has to go through a protein channel. So what do diffusion and facilitated diffusion have in common? Well, it's both moving molecules from high to low. It's both passive transport. You got to know that. Facilitated diffusion and diffusion are both passive transport. They do not require energy. The difference is only that facilitated diffusion has to go through a protein channel. And actually, I want you to write this in. Water can move through a channel called an aquaporin. Water can move through a channel called an aquaporin. All right, active transport requires energy. Okay, so active transport is going to be the movement of molecules that requires ATP. Anytime you see pumps, like pumps calcium in, that's going to be requiring energy. So this process is moving material against the gradient. So it's not high to low anymore. We're going low to high. Some examples would be protein pumps, like the sodium-potassium pump, which is what you see here. You're moving sodium out of the cell and potassium into the cell. All right, can y'all see that? Sodium's going out and the potassium's going to go in. It pumps it in there and requires, see these little ATPs? Electrons can also provide energy, such as how you make energy in the mitochondria with the electron transport chain. More examples of active transport are exocytosis. There's a pump right there. Exocytosis and endocytosis. So trying to get, look at this picture. This is nice. You got this vesicle right here, and now you got this food right here. and you're going to form a vesicle, aka a bubble, and bring it inside. So that is going to be endocytosis, or take it outside is going to be the same thing but backwards. You're going to push it outside, which is exocytosis. Okay, flip your paper over. Just make sure you have this. Passive transport versus active transport. I want you to draw this on your paper. So flip your paper over on the back and draw this. First thing you want to know is that active requires energy. Okay, passive transport goes from high concentration to low concentration, kind of like you're walking down a hill. Active transport goes from low concentration to high concentration. Okay, examples of passive transport are osmosis, diffusion, and facilitated diffusion. Okay, active transport, you've got pumps, and you got endocytosis and exocytosis. All right, I hope that was helpful.